We are broadcasting. Okay, so hang tight.
I just ate candy while we were broadcasting.
Okay, Jenny's gone down.
Hi, I am Hattie Beverly Butts, and the other members of my group are Monique Gurney and Jenny Mills, and we will be presenting on the disease called botulism. Botulism can be a deadly, start over. Botulism is a disease which can be deadly, which is contracted through the release of a toxin through the body's nervous system. Its primary indicators are signaled by the weakening of the neuromuscular system of the infected individual, particularly in the face, eyes, and respiratory system. While botulism is fairly rare nowadays, individuals are more at risk for contracting the toxins through injectable drug use, home canned or home fermented foods, or if introduced to particular raw foods as infants. Clostridium botulinum is the etiological agent of the botulism disease. The damage caused by this bacterium is from the release of the botulinum exotoxin into the neuromuscular system of its host. The Clostridium botulinum species has 7 distinct types, which are generally designated as types A, type B, type C, type D, type E, type F, and type G. These distinct strains of Clostridium botulinum release different types of exotoxins and can be found throughout different regions of the world and throughout the GI tracts of different animal species. Only types A, B, E, and F cause disease in humans. Types A, B, and E result in foodborne illness, with type E specifically associated with fish products. This type of Clostridium botulinum is found in water and water sediments as opposed to land soils or dusts. Type C causes limper neck in birds, and type D affects mammals other than humans. No disease is associated with type G. The cells of Clostridium botulinum are gram-positive and are rod-shaped, as you can see in the pictures. This falls under the classification of bacillus. They are motile, meaning that they are capable of motion. This is due to the peritrichous flagella lining the outer cell wall. Clostridium botulinum is anaerobic, which means that it thrives and grows without the presence of oxygen. When conditions are poor, Clostridium botulinum can form a hard protective coating of spores around its cells. This coating has layers of shielding membranes and protects the essential parts of the bacterium from the poor environmental conditions, which could potentially kill the bacterium. These spores are heat resistant. It produces the neurotoxin known as botulinum, which is considered to be one of the most potent poisons known. Clostridium botulinum most often inhabits soil, water sediments, and is occasionally found in the GI tracts of animals. Something interesting that I found was that Clostridium botulinum types A and B are most often found in North America, with type A being found in the western regions and type B being found in the eastern regions. Some of the scientists that were testing these types found that type A seemed to prefer soils with neutral alkalinity, and type B seemed to prefer soils and water with more acidity. The bacterium can easily populate food sources, such as vegetables and fruits. When humans process those vegetables or fruits which have been populated by Clostridium botulinum, they can create the anaerobic environment needed for sporulation and toxin release. This can happen through inaccurate and unsystematic canning or fermenting procedures, or because of inadequate heating processes. The cause of infant botulism has been linked to the intake of raw or unpasteurized honey. While Clostridium botulinum can cause disease in humans under the right conditions, botulism cannot be transmitted from human to human.
Hi, my name is Monique Gurney, and I will be sharing the slides discussing the signs and symptoms of botulism and botulism transmission. The three major forms of botulism share a lot of the same signs and symptoms. The difference between a sign and a symptom are that signs are visible externally, and symptoms are internal feelings that have to be described by the person feeling them. The symptoms of botulism are double vision, blurred vision, a thick filling tongue, difficulty swallowing, and dizziness. The signs of botulism include drooping eyelids, slurred speech, dry mouth, muscle weakness, and infant signs include appearing lethargic, poor feeding, constipation, having a weak cry, and appearing floppy. The usual time for the onset of symptoms is 12 to 72 hours, and that is dependent on the size of the dose of the toxin. The exotoxin called botulinum toxin travels through the circulatory system until it reaches the neuromuscular junctions in skeletal muscles. The way botulinum works is that it prevents the release of the neurotransmitter acetylcholine, which is what initiates the signal for muscle contractions. The picture on this slide shows the physiological effects of botulinum. As you see in the bottom picture, when the toxin enters the motor end plate and attaches to the presynaptic membrane, it blocks the chemical, preventing the impulse transmission and prevents the muscle from contracting. This causes flaccid paralysis. The neuromuscular systems first affect the muscles of the head and is what is responsible for the double vision, difficulty swallowing, and dizziness. There are no sensory or mental lapse that occur, but later symptoms of muscular paralysis and respiratory compromise can lead to death from respiratory arrest. The fatality rate has been reduced to about 10% due to mechanical respirators and modern medicine. There are three types of botulism: foodborne botulism, infant botulism, and wound botulism. Foodborne botulism is a pure intoxication. Many of the outbreaks occur in home-processed food like canned vegetables, smoked meats, and cheese spreads. There are several factors in food processing that lead to botulism. Endospores are on the food at the time of gathering and are difficult to remove completely. The contaminated food gets into jars and steamed in pressure cookers, but may not reach the pressure and temperature needed to kill all the endospores, and some may survive. The endospores are highly resistant to heat, and the canning process removes air, which creates an anaerobic environment. This favors endospore germination and vegetative growth. It is not always evident that a product has a bacterial growth from the taste or the texture, but it may still contain some of the toxin. Culture and diagnosis of foodborne botulism includes lab work attempting to identify the toxin in the affected food, seeing a trend in people consuming the same kinds of food and having the same symptoms, and the cultivation of the bacteria in feces. Usually the cultivation of bacteria in feces is the confirmation of diagnosis. Infant botulism is infection followed by intoxication. This is the most common type of botulism found in the United States. There are about 75 cases reported annually. The exact food source is not always known, but there are some common sources such as raw honey, as was mentioned before. The immature state of neonatal intestines allow endosporins to gain a foothold, germinate, and give off the neurotoxin. Adults can also ingest botulinum endospores in contaminated vegetables and other food, but the adult intestinal tract normally inhibits an infection. Finding the toxin or the bacteria in the feces confirms the diagnosis for infant botulism. Wound botulism is also infection followed by intoxication, and there are usually only 3 or 4 wound botulism cases each year in the United States. However, the cases are increasing in the United States due to the reports of intravenous drug usage and needle punctures. The endospores enter a wound or a puncture. The culture and diagnosis for wound botulism is that the toxin should be in the serum or organisms should be grown from the wound.
Hi, my name is Jenny Mills, and I'm going to go over treatment and prevention control, and also recent ethics surrounding Clostridium botulinum. There are several different types of treatments depending on how the person is affected. Induced vomiting or the intake of laxatives may be a treatment from someone with foodborne botulism. If someone has a wound infected with botulism, the doctor may have to perform surgery to remove the infected tissue. If you're diagnosed early enough with foodborne or wound botulism, then the injection of an antitoxin may be a viable treatment to reduce the risk of complications. The antitoxin will bind itself to the botulism toxin and keep it from affecting anything else. Unfortunately, it is not able to reverse or treat any damage that has already been done in the body. There are many people who are able to recover fully, but the recovery process is long and it may take months to heal the destroyed nerves. Antibiotics are only a viable treatment for wound botulism. Using it on any other kind of botulism quickens the time at which the toxins are released. If one of the problems of being affected is problems breathing, a mechanical ventilator may be used to help. The main thing that can be done to prevent the infection of botulism is to properly can foods. Pressure cook them at 250 degrees Fahrenheit for at least 20 to 100 minutes depending on the food. It is not a good idea to eat preserved food if the container bulges out or smells bad. If potatoes are cooked in tinfoil, they need to be eaten hot or stored in the refrigerator. Oils infused with garlic and other herbs also need to be stored in the refrigerator. These are the ways to prevent foodborne botulism. Since bloodborne botulism is usually spread by injecting illicit drugs, mostly black tar heroin, it is not a good idea to do this. So an article written by NPR and a shorter version by the Pandora Report has emerged within the last couple of years, raising questions about the ethics of withholding bacterium information. Because this article is so short, we would like to share its contents with you now. We ask you to pay special attention to the issues presented in the article surrounding Clostridium botulinum and ask what your thoughts are regarding the subject of ethics. 2 years ago, Dr. Stephen Arnon and Dr. Jason Barish discovered a new strain of Clostridium botulinum. Typical C. botulinum strains are known to express any of the 7 different botulinum neurotoxins, botulinum toxins types A through G. The new strain, discovered by Arnon and Barish after studying infant botulism at the California Department of Public Health in Sacramento, was found to express neurological toxins, botulinum toxin type B, and a new botulinum toxin type H. Dr. Arnon and Dr. Barish published their findings of the new toxin in the Journal of Infectious Diseases in 2013, but elected to withhold from the public and the rest of the scientific community any genetic sequencing information regarding the new strain. The withholding of this information has remained a point of contention between the researchers and individuals representing various organizations wishing to study the bacteria. Given the lack of an antitoxin antidote available to stop the dangerous effects of the type H toxin, many individuals desire to perform research on the strain of C. botulinum that can produce the type H toxin. Several scientists and federal institutions have tried to request the sequences and/or live strains of Aaron's new strain of C. botulinum. However, Arnon remains steadfast in not sharing the bacteria. The case raises an unresolved issue that persists in the sciences. That issue is defining the parameter by which we are able to distinguish dual-use research. Dual-use research in the biological sciences is research that can be performed to benefit humans but can also be performed to harm humans. In this particular case, the type H toxin has been declared the most deadly toxin and has great potential to be deployed as a biological weapon. The absence of an available antitoxin that can be administered to infected patients raises great cause for concern that the bacteria producing the toxin could be mass-produced to harm innocent people. From NPR's story, it seems that this sentiment is shared with Type H's discoverer, Arnon. Upon Arnon's discovery of Type H, the CDC, U.S. Army Laboratories, and DHS all expressed interest in acquiring the strain that produces this new neurotoxin. These federal institutions' interest in studying the toxin in order to develop a cure is the same goal as numerous other scientists who want to perform research on the strain. So how does one build biodefense against a pathogen one cannot gain access to? Maybe from Dr. Arnon's perspective, keeping Pandora's box closed may be the best weapon of defense for the U.S. against the botulinum type H neurotoxin. So here's a slide of the references used throughout our presentation. We hope that you enjoyed learning about botulism with us and had a good time watching.